Hello, everyone. Welcome back to the MEEP Sciences channel of Dr. Ghosh. Today, we are going to deliver this particular talk as a part of Student Mentorship Program 2024. I, Dr. Ghosh, Associate Professor and Principal Investigator of Microbial Engineering and Algal Biotechnology Laboratory, Department of Biosciences, JIS University, Kolkata, will deliver the talk on a very special topic of microbial biopigments in textile industry. It has been conceptualized by my PhD and master students and bachelor students as well, Ms. Shrestha Debnath, Afan and Binderjit Singh. So let's get started the glimpse of this particular project and presentation. The textile industry has been a very big challenge for the environment since a very long time in many years. Few of them being firstly the contaminated water, waste that it generates, and secondly the chemicals which are used along with it for dyeing purposes, and lastly the amount of energy which has been spent during the process for various heating and drying techniques. The most important issue being the dye which are mostly synthetic in origin and which has been replaced the natural dyes, and they are going to get thrown. So in overall nutshell, what I would like to say, the most of the synthetic dyes which are used in the textile industries at present, they are not only changing the microbial ecosystem in the environment, they're also polluting the water reservoirs. In the search for respective and representative articles, the term microbial pigment was used. Even though the microbial pigments cover pigments and dyes derived from both bacteria, fungi, even in the algal regime. These pigments derived from them are used in textile dyeing process. With the time, the studies regarding these pigments are increased. So in nutshell, it has been showing that how publication trend has been increasing over years, mostly focusing on microbe-derived pigments in textile dyeing, because synthetic pigments which have been used for last few decades, they have a lot of ill impacts in the environment and the human health. Whereas biodegradable microbe-derived pigments are very user-friendly, they are eco-compatible and environmentally benign while they are in use in the different textile industries. So these biopigments are mostly and predominantly are being synthesized and derived and extracted from different bacterial regime, different fungal regime, and different algal regimes. Then what are the bacterial dyes and how are they derived from bacteria or microbial regime? Bacterial metabolites, especially secondary metabolites, are the byproduct of microbial biomass and growth. They are resulted of or in response to the induced stimuli, maybe internal or exterior factors. These secondary metabolites are not only responsible for color, but additional properties like potent antimicrobial activity against different pathogenic infection and different anti-cancer activities. These are not soluble in water but are called as dye rather than pigments because they remain bound to textile materials and form bonds with the available functional groups. Several microbial strains are able to produce pigments as a biopigment or biodye, those who are capable of imparting the color to textile materials. In the cotton or fiber, range of the color that these bacteria produce is very widespread, including pink, red, orange, yellow, blue, green, and so many more. These resulting colors or the biopigments or biodyes depends on the nature of the substrate, meaning that the various shades can be obtained by same pigment on different fabrics, different fibers. That shade also depends on the dyeing conditions, the process parameters. These conditions or the microenvironments are various factors. These are intrinsic factors or the external factors are involved, such as temperature, pH, use of mordant to enhance or alleviate the dyeing process. So this particular slide will allow you to understand the basic features of the bacterial or microbially derived pigments that can be transferred during the dyeing process. Pigment production using certain bacterial regime can be altered by using different conditions. During growth, especially, several bacterial strains are able to produce biopigments or biodyes capable of imparting color to textile materials. These bacterial regimes are being isolated from various natural reservoirs like soil, water, plant, insect, and estuaries. Among many of these important bacterial regime includes Serratia, Streptomyces, Pseudomonas bacterial regime. There are different algal regimes are involved from Chlorella, the Volvox, and even Sinococcus, Sinocystis, Plasmodesmatas. So these are different algal regimes are also there from which the biopigment can be get extracted, which are further processed through the series of channel of steps as a dyeing process. So these are the different bacterial pigments. Those who having different functionalities has been shown in this pictorial presentation. The bacterial pigments not only used in the dye industries, they also have an anti-parasitic, UV protective, antimicrobial, anti-cancer, and antioxidative, anti-inflammatory responses as well. So they are the very beneficial biomolecules as a secondary metabolites of microbial metabolism. Then what are the different types of microbial or bacterial extracts used as a biopigment? Bacterial pigments have a wide range of colors that can be used during textile dyeing, but not all pigments can impart color to material. And most commonly used ones are the proteoglycine, biolacin, melanin as a component. These three different biomolecules are mostly known as antibacterial antioxidative pigments, those having high level of UV protective mechanism. These biolacin is mostly found in as a fungal regime, bacterial regime as well, and melanin is also very potent bacterial biopigment extract. And these are their molecular structure, which are being extracted from bacterial regime, or which are used in the textile industry as a natural colorant. Now let's discuss about the dyeing process, how we can able to make the different fibers colorful by using biopigments. The usual procedure which are involved is in a stepwise manner as explained below. It is a cascade of processes where one step is necessary for the next to begin and follow it. So in the first step, the preparation of solution is very important to ensure the proper dissolution of the pigment. The dissolution depends on the nature of the pigment, physicochemical property of the pigment, usually whether it is soluble in ethanol, methanol, acetone, or chloroform. The preparation of dye baths using acetone, ethanol, or water is considered highly eco-friendly and can lead to the production of sustainable dye baths. In the second step is dyeing for a particular duration, keeping it check the condition like pH, temperature. The pH should always be optimized, chosen according to the type of textile material or the fiber quality and their Young modulus. The cellulose and the plant-based materials fibers are the higher pH is desirable, while for protein-based fibers like wool, silk, acidic dye baths are acceptable. In the final and the third step is washing and the drying phase of the textile fibers after get colored. That has been dyed using biopigment. There are various techniques and methods that have been used for the drying process. So these are the schematic diagram of different phases which are the preparation and the dyeing of textile materials and fibers using microbially derived pigments. First is the preparation of the solution, that is the dissolution of the pigment, adjustment of the pH and the salt condition. Then it will follow for dyeing process, which be highly important like temperature and duration of dyeing, which will allow the retaining of the biopigment on the surface or as a cross-link within the fiber. And finally is the post-dyeing, that is the washing and drying of the fibers when the pigments are get attached or associated with. Now, as I said in the previous slides, that mordants are also playing very important role in the drying process. The mordants are the salt that are frequently used as an additive in the dye bath to improvise the fixation of the natural dyes or the pigments to the fiber. The mordant form complexes with the dye are additionally able to attach to the surface of the fiber, thus alleviating the exhaustion of the dye bath and improving the washing fastness of the dried fabrics. The fabric exhibited different shades and hues as shown in figure. on the right hand side. This was tested with the geosine pigment extracted from the marine Serratia rubidi ram alex bacteria. The lowest color depend and the depth is entirely in the sample was obtained from the CuSO4 salt, while of other types of mordant, the color intensity was satisfying. The color hue strongly dependent on the type of the mordant which are being used. So there are different types of mordant which are used in different kinds of fibers like FeSO4, CuSO4, lemon and sodium bicarbonate. These are the ideal example of mordant which will allow the stability of the dye to the fibers or the fabric which are used to get stained by different biopigments. So biopigments not only having the property to get coloring the fibers, it has also different antimicrobial activities, but this is not our concern or major discussion concern in this particular presentation, so we will skip it. But for your information, this biopigment also having different antimicrobial properties. They can not only use for the coloring of the different fibers like linen or synthetic fibers like coffee tone, satin, dacron or polyesters. They're also having medicinal aspects, which are also very growing interest for the biopigment production by different microbes. So we come to a conclusion and concluding remarks as including some future perspective. The emergence in the studies of use of this bacterial or microbe in textile industries has gained a significant amount of strength in recent researches and lots of interest as well as being developed towards it. Further research needs to get encouraged, especially towards exploring new pigments which are derived from algae, investigating their potential use as a textile colorant, which are more environmentally friendly. When they are getting exposed to the environment as a effluent, they are environmentally benign. There are, however, most endless possibilities and combination of different bacterial pigments to investigate with different types of fibers. So fiber versatility needs to be increased. Moreover, it is also possible to investigate the combination with the other antimicrobial compound. As it was shown, different strains of microorganisms produce some compounds such as for pro-digiocin, but these bacterial metabolites have different ability to dye certain types of textile fibers. So the current research needs to be more focused on certain kind of dyes in combination with different mordants that can be applied on the diverse and broad range of fibers for their dyeing and for the coloring of these fibers. The current limitation of these studies lies in the fact that the majority of the presented research is focused more on the isolation and general application of bacterial dyes, but it needs to be get focused more on finding metabolites as a secondary metabolites that can be extracted from bacteria, can replace the synthetic cues, and that will be more sustainable for the textile industry. So in a nutshell, what I would like to conclude in this particular presentation is the current research needs to be focused on based on different new biopigment generation, the extraction and isolation from more new microorganism and regimes, those who are more stable, more eco-friendly, and they can easily work out for textile fiber colorization in association with the different mordant. The process parameter needs to be optimized in more improving the process in the dye industries, in the textile fiber coloring, and more focusing on new kind of secondary metabolites which can be useful more better colorant or more better biopigment in the future for the textile industries. So these are the few productive work that we have done in last 2 years. We have focusing on extraction and characterization of different biopigments from different algal regime. We have isolated from marine water resources of Sundarban region. We have also focused on different value-added biomolecule generations having therapeutic activities and to develop the system biological tools. We have also identified different dye-degrading lignocellulosic bacteria from the mangrove ecosystem, because while you have been generating different biodyes or pigments, before it gets excreted or it is getting exposed into the environment as an effluent, it needs to go for the final processing that can reduce the dye load before throwing the watery part of the industry. We have also focused on the different microbial consortia development that will allow to break down different kind of lignocellulosic waste biomass for its proper utilization for value-added product generation. We've also developed new microbial assemblages in different proportions that can be used in textile industry with a special emphasis on metabolic engineering panorama for valorization with the essence of bioengineering. So these are the overall milestone that we have established while we have been working in microbial engineering and algal biotechnology laboratory at JIS University. Thank you very much for your attention. I hope you enjoyed this particular session, and more videos is about to come with more interest and more knowledge that I would like to share with all of you. Hope you enjoyed this session. Stay tuned. Please subscribe our channel and do like and share. Thank you very much.